So yeah, this one, it won't be, I feel it won't be a lot about polygenic adaptation. One thing about plasticity, also I think this talk is going to be a little bit like the lectures that I've given before, in the sense that it's some kind of overview of different topics, so it won't be very focused on one study. So I will cite an eminent quantitative geneticist who said yesterday, I want you to get the feel of what's going on and don't pay attention to the details. And for that, I didn't put the details to a large extent. So I hope this will work and you'll be able to follow the general frame of the talk. So since we're interested in phenotypic variation and we're talking about hybridization just before, I want to start with these two species of Daphnia that have been described. One of them, Mike Lynch talked about it during his talk. It's coming from Africa and in Asia, and it's been invading America. This is another species that has been described in a similar environment. It's called Daphnia monaca, except that people soon realized that it's not different species. This is actually the same clone when you expose them or not to predator cues, having some fish or their predators, which can be invertebrates actually, in the water. They perceive and they grow this big helmet. So Daphnia have all kinds of different traits like that, that they grow. They have neck teeth, they have different cones on their head. So this is a pretty striking example of what phenotypic variation you can produce with a given genotype. I'll just go through some of these examples just to illustrate. This actually we've seen yesterday in Subhash's talk. This is this darkening of Drosophila abdomen when they develop at colder temperature. And so obviously when you go in nature, you will see some latitudinal declines of this trait. So you will want to look for genetic variation for that. So people who do common gardens, some others will not. They will just sample and genotype. But it turns out that this is what you get with the inbred line when you place them at these different temperatures. And so you can draw what we call reaction norm of the darkness at these different temperatures. And they're more dark at this lower temperature. So this is thought to be helping them to warm under light the colder temperature. In other cases, you see this discrete variation. This, again, a bit like this Daphnia. This is even more striking. This looks like really completely different species, but these are just wet season and dry season morphs of this butterfly. In this particular reason, I don't really know what's, if there's the advantage of that, but in some other species like Bicyclus, you see these eye spots that are supposed to distract or defer the attack of predators towards this part of the wing, which is not very protected. And in our lab, we've been interested in these microalgae that can tolerate very broad range of salinity. When you place them at different salinities, they will have different shapes, cell shapes, and this color here is the fluorescence of chlorophyll, so they have different chlorophyll content. If you do a PC on these traits on the flow cytometer, where you can measure many individuals, you can see that they're different on different sets of traits, and so you can measure their differences. We've also looked a little bit at these traits that's very well studied in this species, where depending on the salinity they're in, they will have different glycerol content, and glycerol helps them to osmoregulate in the face of very high salinity, which is something that can dehydrate. So this is a model species actually for salinity tolerance and for dehydration in general. Okay, so what's the commonality between all that is that, okay, we've heard about plasticity in the week, but I'll just put some definitions a bit more clearly. It's the ability of a given genotype to produce different phenotypes according to the environment in which it develops or where it or where the trait is expressed. So I want to start by pointing out this is not the environmental variance that people describe in quantitative genetics, this VE. I've heard some people say, oh, more plastic individuals have more VE. This is not what it's about. I mean, VE, of course, includes lots of things. It can include some microenvironmental variation you haven't controlled in your experiment. It can include developmental noise, measurement error. This all goes into VE. Plasticity, it's when you really have some genotypes that you placed across a macro environment, let's say, that you have controlled in the lab, and you measure the response to that. This is plasticity. It could be correlated to the VE or not. This is an empirical question. Once you say that, I mean, this is, you see that it's defined by contrast to something that doesn't change. So what's specific to me, the more I think about this, what is specific and it's clearly defined is something non-plastic. But what's not specific is all the rest that we describe as plastic. So it could be, it could have discrete variation across environment or continuous. It could be a reversible trait that changes repeatedly, or one that's fixed during development. You develop in that environment, you produce this phenotype, or in the other, you produce another one. It could be an organic response, this trait of the leaf or something like that. The beak is changing, I don't know. Or it could be a kind of whole organism trait like dormancy or winged morph of aphids, these types of things. More like history. Then there has to be an environmental trigger. This could be biotic or abiotic. So we've seen like it could be the predator presence that's driving it. It could be temperature. It can be just a simple environment or a composite aspect of the environment. And then it will depend a lot on the type of organisms. An organism that has continuous growth, you could say it's developing throughout its life, so it can modify its phenotype a lot. Something that has an external shell will have more trouble. All these kind of details of biology, they matter. If there is a metamorphosis or not, maybe easier to produce completely different phenotype depending on the environment. So this is all this kind of diversity and may seem like vagueness that makes it somewhat difficult to study, but somewhat it seems like everywhere you look, you find some plasticity, especially when you look at natural populations. Also, I want to point out that it's a phenomenological definition. It's just something that you observe. It doesn't have to imply that it's adaptive. Some people, you say, you cannot call this a plasticity because it's not adaptive. Well, then it's becoming difficult to study because at first you just observe something and then you have to make up either interpretation, which people are making, or demonstration that it's adaptive. And demonstrating it is quite difficult because you would need to compare plastic and non-plastic genotypes, right? And this is not often available. But there are some rare examples where this has been done. I think some elegant studies by Joanna Schmidt back in the days in the 90s where they use these transgenic plants. So there's this very well-studied response in plants that when they're in a crowded environment, they grow higher to get the light. So that's it. I put some adaptive argument to get the light. It's well known how it works. The light they get, the ratio of red to far red, is different when it's light that's been reflected by conspecifics. So they perceive that they're surrounded by conspecifics. But you can turn this off by knocking down this phytochrome B gene. And this is what they did here. They used transgenic plants where they had turned off plasticity or not. And then they put them in densely or not densely populated environment. And so they could assess how they express phenotype that they get, what's its fitness in the two environments without having the plasticity. And then you can assess what's the benefit of plasticity. So they found that the observation that phytochrome-mediated elongation is advantageous in dense stands, but disadvantageous for uncrowded plants. This indicates that this response to shading allows them to develop an appropriate morphology for the level of competition they experience, basically. So they develop the right phenotype in where it's most beneficial. But this hasn't been done so much as far as I can tell. So one of the reasons why at least I got interested into that, because I'm a bit like you, I'm interested in adaptation, how adaptation works. I wasn't really thinking about this, but then when you look at climate change, phenotypic responses to climate change, well, these are some article titles that I think it stopped in the 2010. I didn't get the more recent ones, but there are lots of these studies about phenotypic responses, evolutionary responses to climate change. When you look at them, usually people, when they're trying to either use this animal model or place population in common garden, they find that the evolutionary responses, usually they're quite modest. Sometimes they're in an opposite direction. The changes in breeding values are in opposite direction to the phenotypic change. So this is a bit puzzling. And on the other hand, when you have like one of the traits that responds the most to climate change is phenology, changing the timing of seasonal events like reproduction. It goes across plants, trees, birds, etc. And there, as I said in the previous talk, this is a trait for which we think that it makes sense that there's an optimum. You shouldn't be too early, too late. Well, in this example, I mean, for gray teeth laying dates, when you measure, you can measure plasticity within an individual because individuals reproduce multiple times in their lifetime. So you can take their breeding time as a function of their spring temperature, and you do that repeatedly, and then you get reaction norm per individual. When you do that, and you combine this to the trend of environmental change, you get big part of the actual phenotypic response that occurred across here in the population. So this was a clear example that plasticity contributes a lot to phenotypic responses to climate change. Something else I want to point out is that, you know, especially in the previous talks, I was going a little bit between this work on microbes and works on on animals and plants. And usually people working with microbes, it's difficult to measure other traits than population growth, so fitness. And even beyond that, there are lots of people who are interested in just measuring how fitness changes across environments, because this is, so this can be described as an environmental tolerance curve. Again, a paper by Mike Lynch back in the days. It can be thought as a representation of the fundamental niche, which the fundamental niche, Hutchinsonian niche, is the growth rate of population against multiple environmental axes. If you slice it against one axis, you get something like that, the growth rate. The range where population can grow, this is the fundamental niche, one axis of it. So this is studied a lot especially to predict responses to environmental change. So usually they're hump-shaped like that, you know, they have an optimum environment. What can I say? For temperature, they usually skewed. High temperature causes faster decrease.
What's the difference between environmental tolerance curve and
performance curve?
Reaction norm?
I mean, reaction norm is a very generic term. It's just how phenotype changes with the environment. So you can apply the reaction norm to a trait, and then usually would call that plasticity. For that, I wouldn't call that plasticity, is the only thing. A reaction norm for fitness, I wouldn't call it plasticity because, well, for various reasons. For instance, you tend to think that when fitness doesn't change much with the environment, people say the organism is more plastic, usually, because it's plastic and it's able to tolerate better. But then no change with the environment is not what you describe as plastic. So I find it confusing. More plastic means more change with the environment. So that's why usually I prefer thinking of things in this way, where you have these underlying traits that we're interested in, and this responds to the environment. So you have this reaction norm for the traits, right? These are 3 genotypes in different colors. And so there are different, this one is non-plastic, it's flat, and this one is very plastic, okay? And then you also have this kind of this fitness landscape with how fitness changes with the trait and the environment. So this is just, this 3D graph is just a moving optimum, really. It's just what we've been discussing. So this is like here, along this direction, you have an optimum for the trait, but it changes with the environment, right? And when you place the reaction norm for the traits on that, then from this, you get these tolerance curves, basically. So there's if you just basically follow this red line here and follow its fitness, it will have this curve. And this one, which is not plastic, it will have this different curve. So the position, the position gives you the optimal environment, and then the slope, how it relates to the slope of this optimum, it will provide the breadth of the tolerance curve. So I don't know, you can think about this or look at it, but it's just, I mean, just this idea that there's plasticity of underlying traits which translates into how well you tolerate the environment. It's kind of, I think, a useful way to think about this, and also can connect predictions about the evolution of plasticity to those about the evolution of these tolerance curves, like broad tolerance curves just means plasticity that tracks the optimum phenotype across environment well. Okay. Okay, so this was kind of the introductory part, and now the outline of this talk is that I want to start about general consideration about what's the genetic variation and selection on plasticity, how this works. Then I want to move on with this bigger section will be about evolution of plasticity in the fluctuating environments. This is the main part of the talk, I guess. And this third part, I don't know if I really have time to go into that, but we'll see. It's about, because usually people have thought about plasticity in heterogeneous environment, stable environment, and then what happens in a new environment, like under climate change. What can you say about this? Okay, so starting with this, what's causing genetic variation in plasticity? This is kind of a topic that's actively researched because, I mean, it's thought that most plasticity for most traits depend on gene expression plasticity. And so lots of people are doing this gene expression responses to the environment. But then in some cases you have a character candidate trait that you're interested in, and then a candidate loci, and then you can map them. But in case you don't, and you see just this massive response of multiple traits, multiple genes, sorry, that responds to the environment, the dimensionality is very high. Usually the phenotypic dimensionality is not that high unless we go towards phenomic, which some people are advocating, but this requires having some tools to measure many traits. Actually, you will also need probably different ranges of environment to see subsets of genes and trait responding, but the point is you have this high dimensionality. I should have perhaps mentioned there were historical ideas about genetic variants of plasticity. Is it like some specific alleles are environmentally specific, that responds to the environment, environmentally sensitive versus gene expression traits? Now this view that gene expression is involved seems to be predominant. So of course this dimensionality issue, it's the same as for GWAS. Everyone's trying to make sense of it when you have this gene expression trait. So one way is to use these clusters of co-expression. You can do co-expression across genotypes. You can do co-expression across environments. One question we were asking in this paper we wrote some last year, 2 years ago, it was kind of a brainstorming exercise of how you can use responses of gene expression to the environment and responses of traits to environment to maps genotypes to traits, because gene expression identifies some loci. And then we were moving on towards more evolutionary questions. But one thing we wanted to look at is this co-expression pattern is just something that flattens the thing. It's just some correlations, basically. And what you're actually interested in is more like a dynamic process of co-regulation, etc. And so we started to think a bit about this by using this Wagner model of gene regulatory evolution, where so you have a set of genes, and each one of them, so you have this gene that produces these proteins, let's say gene products. Each of them regulates each of the genes, so they're kind of transcription factor. There's a matrix of how each gene products regulate expression of all the genes. Then you iterate that until you reach some equilibrium of for this component. And that the simple question we ask is, what if you introduce, there could be different perturbations that the environment does to that. It could, when you change the environment, it could change the initial conditions. It could change the initial quantity of these particular gene products. And then you iterate all that and it will affect all the composition of the protein content. Or it could have a sustained effect. The environment is just pulling this particular gene product to higher frequency in the cell, and then this will have downwards effect on all this, cascading effect on all these other genes. Or it could have an effect on interaction strength. Basically, it hits one of these elements in the matrix, And you can think of this as the environment targeting, changing the regulatory, I don't know, the methylation close to a particular gene, and then it's not regulated in the same way by others, right? So we did things where we did these types of perturbations, and we looked at what this causes for the correlation matrix that you get of this expression of all these genes, when the true network is that. So a red arrow is inhibition, a black arrow is upregulation, I guess you say, and they can self-regulate, etc. Well, turns out we had kind of trouble finding situations where these different types of perturbation led to different correlation matrices. So, for instance, this one is quite similar to this one. You have this pattern of negative. You have these blocks of positive regulation, etc. But in some cases, you could find some very drastic differences. So, I mean, this was just a starting point. I guess these guys, they work much more on this, and they will follow up on that to try to see if we can make sense of this co-expression patterns in more functional way, let's say. But that's all I want to say about gene expression, I guess, or the functioning of plasticity. Apart from that, I'll stay in this very more statistical description as classical quant gen. So if you think of now the reaction norm for a quantitative trait, so a continuous trait, so you place the phenotype, so it's the expected phenotype, so it would be a phenotype of a family or something like that, across environments, and you have this tolerance, this reaction norm, sorry. How can I get rid of this? Okay, so it did work. Let's see if I can make some other things appear. Okay, so you can think of this in two ways. For instance, it may be that you have just measured things in this and that environment. In some cases, really discrete environments, you have two different host plants for an insect. In some cases, just you have these two treatments. Control treatment, you call that control and treatment, but the organism doesn't care for it, it's just two environments. So, and then you call this is trait value 1. This is another trait, it's trait 2. You could think of this, the same trait measured in two environments, wing size in high temperature and low temperature. You could say it's two different traits if you want. But then they can be genetically correlated. If they're strongly genetically correlated, 100% correlation is the same trait, basically. Otherwise, no. Another way to study that is to focus on the shape of that and say, oh, this intercept here and this slope, these are different traits that may vary genetically, and selection can act on this. And this is what I'll be talking about the most because it's kind of a simplest way to model plasticity in response to continuous environment, where the slope here quantifies plasticity, how much the trait changes. Okay? Then we've seen graphs like this before, so the traditional way of thinking about this, when you have these two, for instance, two environments, is that, so these lines here are different genotypes. So when it's a sexual species, you can have this, you have families, so you do what's called a split brood design, I think. Bruce can confirm that, I guess, or you put the offspring from the same family in different environments, basically, I think. This has been worked out a lot by Sam Shiner, in particular. So if they have the same response to the environment, there's no mean plasticity. Here they have positive mean plasticity. But then if the lines don't have the same, they're not parallel, basically there's genetic variance in plasticity. They don't have to cross, but as long as they don't have the same slope, so this is equivalent to a G by E interaction in these types of formulation of the model. Lately, so we've been thinking with Pierre de Villemereuil, so you've seen his face previously. He was a former postdoc of mine, and now he's one of the co-supervisors of Isabella, actually. During his postdoc, we were starting to think about how can you quantify how much variation there is in shape, you know. Is this reaction norm more linear than curves or these types of things? Does this vary more? Because there's been a meta-analysis on that, to say it shortly, wasn't really, they didn't do things correctly. They had, I mean, it was, I won't go into that now, but we want to try to proper way to formalize that. And so we came up with this this partitioning of phenotypic variation for reaction norms across environments. So again, this is kind of work in progress. It's on the repository. But basically, the idea is just to, if you get the mean phenotype in each environment, from this you get the mean reaction norm, and you can have the variance that's caused by that. So it's, Pierre would argue this is not exactly the same as environmental variation versus it. I'd have trouble explaining it, but the point is that we have these different components where this is the mean trait across environments, and you want to see how much is varied. Then when you remove that, you're left with the genetic component, which you can decompose as usual into additive and non-additive. But here, part of the non-additivity comes from the non-additivity of reaction norms, actually. And then I'll explain later what I mean by that. Etc. And then you can decompose this into okay, the usual additive variance would be like if you take for each genotype its mean across environment and you take the variance of that. But then you have this additive by environment interaction. And then what we're doing is to quantify the contribution of different components of reaction norm shape to the overall mean plasticity and to genetic variance of plasticity. For instance, for this reaction norm, we can say basically, well, the proportion of the phenotypic variance across environments that's caused by slope is 33%. The proportion that you can attribute to curvature is this. But then you can also have how much the variance in slope contribute to genetic variance of the trait. So this might seem like it's a bit detailed, but actually lots of these models that I've used and lots of people have used in the theory, they just assume linear reaction norm, genetic variation in that. And it's just an open empirical question to what extent you can, over the range of environments that are relevant, you can consider that genetic variance is mostly in slope or mostly in curvature and things. So that's it. And then you have, okay, I'll just skip that. One point is that, of course, the contribution of plasticity to the overall phenotypic variance is kind of, and also the contribution of different shape components will depend on your range of environments. This is one thing that makes it difficult to study plasticity. If you have this range of environments, you would find a strong contribution from the curvature, whereas here you'll find that the slope is largely causing the variation across environments. And one of the
So I'm trying to think about how this differs from random regression, where instead of just looking at the reaction norm, you take the reaction norm and then map that into the environment and get a specific.
Yeah, I think it's this. This is based on random regression, but then we're really interested into getting these different contributions of different shape components. It's just some metrics that we came up with, so it also works when reaction norms are nonlinear in the traits, in the parameters, such as this type of tolerance curve function that I showed, where it's not a linear model. Basically, you have products of the parameters and things. So again, I'm not going much into the details of that. We're just trying to make an R package of that. But to me, this has been a very productive discussion in the sense that I was coming from theory and I was trying to find the metrics that we see in theory, and it wasn't really how it's done in the statistical methods I had seen. Particularly in the theory, as I'll show later, you have something that comes up, which is the derivative of the phenotype with respect to its parameter, which will depend on the environment. This is a key component that we're using here in this.
Could you show the equation again? In the environment, the original environment and the new environment? The E's, the epsilons, aren't they epsilons? Yeah.
This is not the original environment. It's just some place where where it's centered. Basically, it just determines where this is centered. This is just something that's it's just something to create. You have a center position parameter, then you have an asymmetry parameter, etc. You don't necessarily have to pay attention to the whole thing. It's just that you can estimate contributions of these different parameters to the overall genetic variation of the trait across environments. This is the point. And you can do it in models where you cannot apply linear modeling. So in this case, so the idea is then to use random regression. It's been pointed out by other people that, but except that this can also be nonlinear random regression, which makes it a bit more general, I guess. For more details, ask Pierre. I mean, I shouldn't say that. I was at the incentive of that, but I'm not really a stats person, so I also have some difficulty trying to really explain the difference with function value traits. I mean, it involves, we're using things that relate to things that already exist, but we're doing it a slightly different way, I guess. And our aim was really to find genetic variation in different aspects of reaction arm shape. How do you compare that, considering that they're in different units, right? The curve is in trait, I don't know, trait squared by environmental squared, etc. That's the thing with plasticity traits, is that they have units of traits and environments. So it can be a bit tricky. So we use everything in terms of the phenotypic variation. Okay, anyway, so going back to the main thread, now you have these different approaches to measuring plasticity. You can think of these different traits in these different environments. This is called the character state approach. So this goes back to Falconer, and it's been popularized in evolution by this Vienna Landy paper. And then you have this more like what we call curve parameter approach. Some people call that reaction norm approach, where you have these traits that are the shape parameters that I just talked about before. Turns out that in both cases, if these traits, either the trait value in each environment or this curve parameter approach, the curve parameter traits, they're polygenic and normally distributed, then you can apply the multivariate breeders equation to them, which we've seen before. So the change in the mean trait, but this is a vector, as I think Enrique was presenting yesterday. You have the vector of all these traits. So it could be either the vector of trait values in each environment, or it could be the vector that includes the slope, the intercept, the slope, the curvature, etc. The change in this trait equals the directional gradient on this trait times the genetic covariance matrix of these traits, right? So in one case, the genetic covariance matrix is the variance in each environment and the covariance across environments of the same trait across environments. In this other case, it's like variance in slope, variance in intercept, covariance between slope and intercept, etc. You can switch from one to the other, depending on what's more convenient, what you have measured. People who work on, these natural populations, for instance, they don't have two environments. They have usually a continuum of, they didn't control the environment, so they use this, they really estimate intercept and slope, and they're interested in that. People doing experiments, usually they will have this control and treatment, and so they prefer this approach. But once you start having multiple environments, then you can choose. Okay?
You don't need to have a linear reaction norm. I mean, random regressions allow you to have that as arbitrary as you want, just a degree of polynomial.
Yeah, it works for polynomial. It works for many different types of approaches, as long as yeah. Okay, so one key things now, if you think of response to selection, is that usually there's no direct selection on plasticity per se, or if there is, it's something specific, what people call the cost of plasticity, for instance. Is being plastic something that affects your fitness regardless of the phenotype you produce? But apart from that, usually what we think of selection of plasticity as selection from plasticity arises from the fact that it causes the genotype to have different trait values in different environments, right? And so it arises from selection on the trait. So, and then for a given parameter theta, the intercept, the slope, whatever, the selection gradient, so you can find the selection gradient on it, and it's just this chain rule that we discussed. So it's the selection gradient on the trait times this thing that you could call the reaction norm gradient, which is how the mean phenotype changes the derivative of the phenotype relative to this parameter, for instance. So this is kind of implicit in the theory, but it comes in the computations actually. So you might as well give it a name, the reaction norm gradient, if you want. And this depends on the environment. So for instance, if you have a linear reaction norm, just as I showed, and I sort of always use that as an example, So you see that, by the way, I kept this E term, the residual component of variation. So this is really the, like the heritable component, I don't know how to say that here, the breeding value or genetic component if you want. So if you have this linear reaction norm where B, the slope, quantifies plasticity, then this derivative relative to the intercept is 1, the derivative relative to the slope is the environment actually. So it's like you reverse the thing and you have to take the derivative of the reaction norm relative to its parameters. And this is what we're using a lot in this approach I described with Pierre. So we base lots of things on that. And so then the selection gradient vector on this reaction norm, selection gradient on the intercept and slope, is just selection on the trait times 1 for the intercept and epsilon for the slope. So selection on the slope, selection on the trait multiplied by the environment. So there are some tricky aspects as to where you define your intercept and things, but let's not go into that. Okay? So these were just like the basic things that you need to go to the next step. So is it like, is it clear? You have questions about this? Okay, if not, then I'm going to go to what happens in a fluctuating environment. Okay, so this is something I've been interested in for some time. So, and lots of people have worked on, but so if you think of what plasticity does when you have a randomly fluctuating environment that's stationary, it fluctuates around the same mean, etc. What reaction norm slope really does is that it really affects the variance of deviation from the optimum, right? The optimum is fluctuating and then the trait is fluctuating itself more or less just because of plasticity. And this will affect, it will not affect the mean mismatch with the optimum, it will affect the variance of this mismatch. And so one kind of simple idea is that plasticity will buffer the impact of the fluctuating environment. Only the environment that induces the phenotype can accurately predict the future selective pressure, where the optimum will be somehow. So otherwise, plasticity, if there's too much plasticity or in the wrong direction, it will cause phenotype to respond too much. It will overshoot the optimum a lot, for instance. And this can amplify fluctuations in fitness. It may cause extinctions. I didn't cite, this is a paper by Tom Reed in 2010, I think. They just had this very simple model with a given plasticity, given slope of reaction arm, and the same variance of fluctuations, but these were like weakly predictable. It was like white noise, and this was highly predictable. And then here, population underwent these very massive fluctuations because they were very often maladapted, and some of them went extinct, whereas here, plasticity sort of buffered this effect of fluctuation. Okay, so we've been trying to, so this is a very simple idea. As usual, these models, they have simple things, but it's very difficult to test in nature. So I'll go back to this study that we've done also with Pierre when he was up with me, where we're trying to estimate fluctuating selection on breeding time in birds and mammals. So I didn't show it again, but we had this 30-something species studies. And so, yeah, the idea that I mentioned last time, so that of course you have this, this red line here is the fitness function. So this optimum might be fluctuating across years, but the phenotype distribution might also be fluctuating. If you're thinking of evolution, this might not be drastic fluctuation, but when you're thinking of plasticity, this can be very large actually. And lots of things will depend on that, what I showed earlier, population dynamics, but also the selection. If you're just measuring a selection gradient, the slope will depend on how the mean phenotype deviates from the optimum. So the fluctuating selection that you will detect using this selection gradient that people use, it will be influenced by that. And so the variance of this mismatch with the optimum is simply the variance of the optimum plus the variance of the mean phenotype over time, variance of the mean phenotype. minus twice their covariance. This is just the variance of difference. So just this says that this variance of mismatch could be buffered if this covariance is positive. This means if there's adaptive tracking of the optimum, right? If it's negative, it means when the optimum is going this way, the phenotype is going the other way, right? But if it's positive, there's some tracking going on. And so in this study, as I said, we had individuals that reproduced several years. And so we didn't have the environment per se, but we had the phenotype of an individual, the breeding time in different years. And so our assumption was that when they changed their breeding time, they did that because of plasticity in response to the environment. Could be other reasons. It could be senescence. It actually occurs that older birds, they reproduce later in the year, but we use that as so difference in trait in breeding time between individuals that reproduce in subsequent years, and we estimate the correlation of that between that and the movements of the optimum that we infer from the fitness trait relationship. And we did that across species, and we found that in several of these species, this correlation was significantly positive. It was also overall significant for birds, significantly positive. So this means that this plastic tracking of the optimum across this bird species, it buffers, it reduces the magnitude of this fluctuation in the mismatch with optimum, which hadn't been shown before. Even though this is like the idea, this is why plasticity is thought to have evolved in this species. Okay, now if you think more in evolutionary terms, now if we go back to what I showed you before, again, this linear reaction norm, now we're going to be a bit more specific. This environment is, I call that the environment of development, is the environment that will have an influence of the trait. It may be that the temperature is affecting the trait, but it's affecting it at one time where it can modify the trait, and then there's a delay where the trait is produced, the response is produced, and then the optimum phenotype may also depend on the environment, but sometime later, of course, because selection might act later on this trait. So there's this, it may also be another environment, but let's keep it simple for now. Let's say it's both temperature, but at the time of development and at the time of selection. And the response to selection in one generation for this reaction norm vector, which is here the intercept and slope, you can use this Breeder's equation. So it's G beta. So this G matrix has on the diagonal the variance of intercept, variance of slope, and then the covariance here. And then this beta, as I told you, it's equal to the selection on the trait times this reaction norm gradient. And now the selection gradient on the trait depends on the deviation from the optimum, so you can expand these terms that depend on the environment, right? And so you have this difference between the intercept and the optimum in the average environment, let's say, put it this way, minus this difference between the plastic response to the environment of development minus the optimum change with the environment of selection. Okay, makes sense? See what I mean? Okay, and then you can multiply, so replace this by this, you multiply, and then you can take the expectation of that when the environment is fluctuating. So there are stochastic fluctuations in the environment. And so when you do this, you end up that this expected selection gradient will depend on A minus A bar for the intercept. These E's cancel because they're assumed to be centered, let's say, to have a mean of 0. Whereas for plasticity, you're left with this term. So the expected selection on plasticity. So this cancels when this is 0, right? So this means that B is selected towards this optimum plasticity, which is the slope of how the optimum changes with the environment, multiplied by the correlation or the autocorrelation here of the environment of development and selection, right? So there are different flavors of this. This could be different environment. Actually, the real thing is a regression slope. It should be a regression slope of the environment of selection on that of development. But when it's the same environment at different times, it becomes an autocorrelation. This is just a detail. Okay? So plasticity should evolve to the slope of the optimum, discounted by this autocorrelation. I mean, reduced, because we expect the autocorrelation to be below 1. It could actually be negative. And then you also see these terms coming out here. So this means that selection on the plasticity is not just proportional to the strength of stabilizing selection, it's also proportional to the magnitude of fluctuations. Bigger fluctuations, they will cause faster evolution towards this equilibrium. Okay. So this is a result that came in different modes, in different ways. You can also have something about Q reliability. I mean, but it's kind of the same idea all the time. There's this predictability issue of the environment. You can also think that in nature, usually organisms are responding to complex environments that have multiple axes, and we don't know what these are, so we just measure temperature as a proxy or something, but that's not what they see. The birds, it's known that they use a combination of photoperiod and temperature to sort of know which season they're in. Oh, it's cold for spring now, or something like this. And other things like that, so combination of moisture and temperature. So you can do that. I mean, these models is very easy to do that. And actually, it was the case in the original model by Gavrillets and Scheiner, where they had this multiple traits responding to multiple environments. They kept it a little bit abstract, so we sort of worked out to understand a bit better how this works. So in this case, you have reaction norm in response to multiple environments. We have environment 1, environment 2, and it becomes this plane, you know. And then the reaction norm plasticity vector, it just gives the direction of biggest slope in this plane. So you could call that the Q, the phenotypic Q. Multivariate Q is the combination of environments that causes the biggest plastic response in the trait, right? If you change a little bit of this one and a little bit of this one, you get the stronger plastic response. So this is kind of the multivariate Q that evolves. Now, if you have a fluctuating environment, you can model like this multiple Qs over time and how they're correlated among each other, etc. There might be a combination that causes movement of the optimum and another one that causes movement of the trait, actually. And so the equilibrium Q that evolves is the linear combination of environmental variables that best predicts the movement of the optimum, even if these movements of the optimum are in response to other variables, other combinations. And so this is like a multivariate thing. And then as usual, when you project something multivariate on just one axis, you can have some an intuitive result. Like for instance, plasticity with respect to specific environmental variables can be not just in excess or opposite to changes in the optimum, but they can be different from what you would expect if you just measure this variable, the environmental variable 1, let's say it's temperature, and you measure its predictability. You say, oh, this should be the level of plasticity scaled by the slope of the optimum. This should be it. And for the gray, it should be that. But actually, because of how these variables are correlated among each other at a given time and across time, the plasticity that evolves is, for instance, steeper than the slope of the optimum or opposite to the slope of the optimum and different from what you would predict based on just this environmental variable. Still, the full multivariate Q is still adaptive. So this is just one of these numerous declinations of I don't have all the traits, you know. Here is I don't have all the environments. But it might be, I think it's good to try to think about this when you're studying plasticity, either in the lab or in the field, and you just follow the few environmental variables. This I would skip. So what's discussed a lot in the context of climate change is that there's thought to be changes not in the mean environment, but in the patterns of fluctuation of the environments and in the predictability and correlation structure of environments. And so if there's a change in Q reliability, that's thought to be the case a lot. Some people describe this, you know, ecological traps. I don't know if you've heard of that. Like the birds, they get into urban areas and they get wrong cues. You know, they misadjust their phenotypes because they're not just they get the wrong information. So if you have a change in the nature of these cues or their reliability, this can increase the variance of mismatch with the optimum when it's fluctuating, as I just showed before. And this could cause extinction risk by itself. And then evolution of plasticity may then be required in this case for evolutionary rescues. For instance, this is what I had represented in this scenario. Like, this is a bit extreme. Like, the predictability switches from this to that, 0.2. So now you have too much plasticity initially. And so the population here starts to crash for the reason that the mean mismatch is not changing, but there's such a variance of mismatch that decreases the expected growth rate of the population because it's never really well adapted. So it starts declining, but then plasticity evolves to this level that matches the level of predictability, and then the population is rescued. So this was just an example. Then we studied this in a bit more detail in that paper later. Okay, something else that we might be interested in is what happens for QTL that affects plasticity, or put it another way, the QTL whose effect depend on the environment, which is the same thing. So QTL with the environmental dependent effect. So I don't know if there's a name for that. Do people study that? No? You don't know? You don't know the name? Is there a name? Classic? Plastic QTL, yeah, but I don't know if there's if it's a thing, you know. For the variance, yes, so it's different, yeah. So, unless it's the variance across environments you have to control. So, you know, I showed you this model last week where for a classic QTL, if you change the variance of fluctuations or the autocorrelation, it won't affect the trajectory of this QTL, at least measured in this logit. Yes? Yeah, probably, yes.
That would be G by E.
Yeah, true. Except that, yeah, G by E is just measured in terms of its contribution to variance, but here you really want to measure its effect on the trait.
But people map QTLs for G by E.
Yeah, it's true, yes. Yeah.
So a lot of work is in the agricultural literature on mapping the QTL by E interactions. And the key there is you need very predictable environments, which is what you don't get in.
You were working on that too, no? You had some things about that? Yeah, the key is the environment that we predict. Oh, in your experiment, yes.
So things like growing regions and stuff. So Luis Gutierrez has done a lot of this, for example, mapping QTLs for wheat production in Uruguay. And the idea there is you have predictable features of the environment, such as location. You have unpredictable features, such as year-to-year weather. And so you can remove some of the variation on the predictable part, but not the unpredictable part. And that's the problem with plasticity, is you basically have to, if you can predict something about where the environment is going to go, you can work with them in terms of evolution. If you can't, then they may actually be more harm than they're worth.
Yeah. Okay, thanks. So anyway, the pattern of fluctuations does not affect. If you have more variance in the environment, it doesn't expect the mean selection coefficient. Here I mean fluctuations of an optimum phenotype, okay? Whereas for a plasticity QTL, it does actually. So you change the predictability of your environment, or just the variance here in this simulation, I just changed the variance of the environment. If you remember from a previous slide, I said, from a quantitative genetics perspective, without looking at loci, how strong selection is on plasticity will depend on the variance of the fluctuations. And here, the expected trajectory of the mutation that causes a change in plasticity, if plasticity is suboptimal in the genetic background, and then you have mutation that's higher to what would be the adaptive plasticity, the most adaptive plasticity in this regime of fluctuations, then just changing the variance of the fluctuations will, increasing the variance of fluctuations will increasing the mean selection on this plasticity QTL, for instance. This is a bit complicated, but or not. I find it interesting to think that there could be a QTL that has pleiotropic effect, so it affects the trait in the average environment, and it affects its plasticity. And what if so the mean background is at the average optimum. So there shouldn't be a benefit to have a mutation that moves you away from the optimum. But through its beneficial effect on plasticity, the mutation can still spread. The mutation spreads, and it has this pleiotropic cost that it shifts the optimum on average from its environment, but it helps this genotype deal with the fluctuation aspect. And so you can have this situation where then the mean background evolves to compensate for the pleiotropic cost of having this plasticity QTL that carries the pleiotropic cost in the average environment. A story. I don't know if you can test that empirically. Think about this. Okay, this was a kind of overview of some of the topics that I thought were interesting about that. And so now probably I'll just finish with this actually, with these experiments that we've been doing in the lab with this microalgae, Dunayella salina. So it's this cute little flagellated phytoplankton, which is a green alga, but actually it can turn, like you don't see it, you see a little bit here. There's a bit of orange, but can fill its cytoplasm with carotene at high salinity under high light. And it contributes to this red color that you see in salterns. This is where they produce salt. It's actually used a lot it's used for carotene production. Lots of this, there's places in Australia where this salterns have been transformed in places that produce carotene with this algae because they have a high carotene content. So it's well studied for this reason. Another reason why it's well studied is like it's the most halotolerant eukaryote. So it can grow from fresh water to saturated brine. And the mechanisms are well understood. So there's this, they produce this glycerol that I talked about. So it's also studied as a model organism for salinity tolerance. Then it's common worldwide in these shallow waters that are very salty. So their salinities, I think, fluctuates quite a bit with precipitation, wind, and sunlight, etc. It's not like in the sea, of course. So you could argue that because it's an extremophile, it doesn't have very many interactors in its ecological niche. So when you're studying its environment, you're modifying salinity, temperature, these types of things in the lab, it captures a big part of this true niche. Of course, when you go down in salinity, then it's encountering many more species. Okay, and generation time is pretty long for a microbe, but still, I think you can do experimental evolution with them. So on the microbial trade-off, I mean, it's a microbe, so you cannot really do things that you do with animals and plants, measuring it. It's a bit difficult to do things at the individual level. But you can do experimental evolution over hundreds of generations. On the other hand, it's still more appropriate than bacteria for measuring traits of individuals, for instance, with the flow cytometer. So we did experiments where we had this liquid handling robot, this pipetting robot, and we drew a random time series of environments in Mathematica, and then we computed what would be the salinity, the mixture of very hypersaline and hyposaline that I need, the volume of that. And then we gave that to the robot, and it pipetted everything. And so this allowed us to have high replication and complex fluctuation pattern, because I was used to this model where there is Gaussian process of the environment. And all I'm seeing in the literature usually is high, low, low, high, and so I wanted to have more something like this continuum. Perhaps it's not needed, I don't know. It made more sense to me, also to connect to the models. And so we exposed them for several months to these treatments. So these treatments is what I showed in previous slides, in previous talks. So is this autocorrelation treatment. So within the treatments, we had at first, for each, we had different strains, and each strain had like 40 replicates. And they had the same mean invariance, but within a treatment, they have different autocorrelation. It could be 0, like this. It could be a very autocorrelated or medium autocorrelation, but positive or negative. So predictability is increasing this way because here it's predictable that if you're somewhat predictable, if you're in higher than average salinity, you're going to go to lower than average salinity. So I should point out, because I don't have a slide on that, that Enrique has done experiments where it's like negative autocorrelation, right? where it's predictable that you go from very high to very low hypoxia, and they found evolution of maternal effects in this case. Okay, first thing we did was to track, so I know we're not here to talk about population dynamics, but that's the easy, low-hanging fruit, I would say, is you measure the population size at each time point, and then you track the population size over time. And you have many, many measurements. We see in a shaded line here. The first thing that jumped to the eye, so they reached this kind of stationary distribution after some time. And this, when you place it on the log scale, it looked a lot like what we were predicting from the theory of fluctuating optimum, you know, that I described a little bit. Yeah.
Again, with the correlation.
Yeah, okay, so the colors are red is we're trying to use this like red noise. So red is the highly positively autocorrelated. The red is negative autocorrelation. So, and the green is no autocorrelation because we couldn't make it white for white noise. Yeah. And so it looked a lot like this. This could be by chance, you know. But it's what we would predict if there's an optimum, as I was saying at the end of the previous lecture. And so you have this skewed distribution. You cannot really grow better than staying at the optimum. But here there's also density dependence. There are lots of. And so try to make it more explicit. We looked at this kind of salinity tolerance curve, which, as I said, will occur if there's some plastic traits and the moving optimum also. And so, but except that actually the way we could capture these responses was when we let this tolerance curve or this growth rate as a function of salinity depend on current, but also previous salinity. And if you just do this quadratic thing, but with dependence on previous salinity also, and these are treatments that you see in the bottom here. So this was highly autocorrelated treatment. This is like negative, you see? These are how it looks here. And then we projected the growth rate that we got on here. It's pretty noisy, but still we were able to fit this, this curve. And now we found kind of a strong effect of environmental autocorrelation on the mean population growth rate, on the variance of growth rate that you get from this in these fluctuating environments, even on other, like the skewness, etc. It fit less well. But the point was like, this is like the straw man a little bit, but if you assume that population growth just depends on current salinity, at least with this quadratic thing, you predict zero effect of environmental autocorrelation on the mean, on the variance of the growth rate. Whereas if you account for the effect of previous salinity, you do. So basically, we call that phenotypic memory because we didn't want to say it's transgeneration plasticity because people would say, oh, it's not generation, it's a microbe. And of course, they transmit their cytoplasm content, everything. So phenotypic memory, their state basically depends on when they have been in few cell generations ago. It's a kind of persistent influence of plasticity. And we think there's a contribution, strong contribution from this glycerol trait. Unfortunately, we couldn't use this trait a lot. We never managed to do this high throughput that we wanted to do, so we just do this glycerol test, you know, it was a bit painful. But so these continuous lines, they're coming from high salinity. And then we place them in high, medium, or low salinity. Whereas this dashed line, they were coming from low salinity, and then they went to low, medium, or to high where they died, okay? And so what you first see is that when they came from high salinity, the first measurement we did of glycerol content, which was just a few minutes after, they already had different glycerol content. They already had adjusted. And the reason is that they're able to excrete their glycerol in the medium. Because if you place them from if they're really filled with glycerol and you place them into very low salinity water, they might burst actually, because too much water comes in. Whereas here, coming from low salinity, they need to produce this new glycerol, so it takes more time. And basically, they weren't able to do it in the high salinity treatment. So we think this is what's explaining this through that you see here. When they're coming from low to high salinity, they suffer a lot because they're not able to adjust their glycerol fast enough. So there's this, here we already have something that's really not like the modes I've been talking about, where the trait is determined once and then under selection. Here, the trait is dynamic and it's asymmetric, depending on the changing environment. We're also interested in looking at fluctuating selection, just like in the model I showed you at this loci, except that here it's just like these two strains that we put in competition. So some of these treatments, they were starting with one strain, and we're just hoping that there would be no de novo variation or residual standing variation. But we also contrasted that with treatments with mixtures of strains. And then we were able to track them by sequencing some characteristic loci. Then we track them, we sequence them at different time points. And so this is the frequency of one of the strains. So at constant salinity, it's outcompeting at low salinity. But then as you increase salinity, it's less and less advantageous relative to the other strain. And these are what happens in our salinity treatments, which is fluctuating treatment, which is quite messy. But anyway, this is a same type of transformation that Nick was showing, where to represent these logit frequencies. Turns out that these types of, just like I was showing in this model that we worked on the mean invariance on the logit allelic frequency, this naturally lends itself to analysis in GLM by logistic regression, which is really the same thing. You're looking at the logit frequency, and then if you put an effect of time, it would be the mean selection coefficient. If you have so it would be, I guess, again, also a random regression. If you have a random effect on the intercept, then this is showing how much this variance increases over, how much the variance trajectory increases over time, and this is about the variance. This is what variance in selection produces. And so, anyway, the mean selection coefficient in the fluctuating environment, the mean slope, if you want, of the frequency change, is lower when you have fluctuations than in a constant environment that has the same mean. The fluctuations reduce the expected selection. And this is, if you remember a slide I showed a bit earlier, we expect that fluctuation will only have an effect on the selection of the mutation if this mutation affects the plasticity of some traits, not if it has the same effect in all environments. So basically, we took this to mean that these different phenotypes, these different strains, they differ in their plasticity or in their tolerance breadth to this environment. It's also consistent with the idea that just like this Jensen's inequality, that if there's a downward curve or concave relationship between selection, between salinity and the selection coefficient in fixed environments, then the average in a fluctuating environment will be lower than the value in the average environment. This is Jensen's inequality. So, well, I mean, this quadratic model was better than the linear model. We just had 3 environmental points, but it seemed to still be concordant with that. I think I'm going to, well, all I want to say about this is that, so if you look at this variance component that I told you, this kind of random intercept to see how these trajectories diverge over time, in the constant environment, we expect this to be caused by drift, by this kind of micro-environmental variation. Perhaps in our incubator, some places are better and they grow better, they have better. So this micro-environmental variation will cause variance among replicates. But the fluctuating ones also have the effect of fluctuating selection. So we found faster increase in the variance of allelic frequency in the fluctuating treatment, but no clear effect of the autocorrelation treatment. Okay, and so another thing we looked at, then this was in the early phase of the experiment, then we went on for a year and a half actually doing this transfer twice a week. And then after that, we acclimated them for 10 days in the same salinity to remove, this is like a common garden somehow, to remove the effect of the last environment they were in. And then we placed them at high and low salinity. And we use high throughput phenotyping, so this time is not as meaningful a trait as glycerol, but we have some ideas about what I showed you in one of the previous slides with cell size, which is this forward scatter trait, something about cellular content, granularity, transparency, etc. And then this fluorescent in red is the chlorophyll content. And so I showed you before, there are plastic responses to salinity. So they have different shape, different chlorophyll content at high versus low salinity. And you can take all these traits and just take this Euclidean distance here, which is how much overall they change across salinities. And when we do that for all these lines that we had, and we plot this against the predictability of the environment, which is a squared autocorrelation, which is how well the one environment predicts the next, basically. We got this kind of clear relationship that was it was the same across these two initial strains that we had on the experiment, where the ones in highly predictable environments had the same plasticity level as the one that stayed in constant environment, which we interpreted as plasticity is not costly because it was not costly per se because it wasn't lost in the constant environment. However, it was reduced in environment where that predicted that evolved fluctuated unpredictably. This goes back to what I showed in one of the slides, that the variance of fluctuations influences the strength of selection on plasticity. So if you're selected towards this, perhaps this also has a cost of, perhaps there is a cost of plasticity, but imposed strong fluctuations are not well predictable, cause a decrease in plasticity. So I have to say, since each time I present this, I don't really know what to understand about this because this is so different from the model predictions where it's for traits that are determined once and then exposed to selection. Here it's a very dynamical trait. Actually, this is my next slide's about that. So if you place them in a new salinity, coming from this acclimation step, and then you track their trajectories over time, you know, in different salinities, in this morphospace, they just travel a lot, you know. Their phenotype moves quite a bit. So it's not evolution. We've done the same with clones and things. It's just plastic changes, basically. And this plasticity, so if you take the difference between lines in two environments and you measure this Euclidean distance, this plasticity, so it depends on the day, you know, after. So our interpretation was like initially you have this, at day one, you have these big differences in environments. It's kind of this passive or very rapid response. Let's say they shrink under high salinity because they lose their water. And then they mount all these responses that involve gene expression and things. But here the gray area is the exponential phase where the population is growing. This is where most people would measure their microbes because it homogenizes things you measure in the exponential phase. Well, they're not very plastic in the exponential phase. They don't feel difference in salinity as far as we could tell in terms of their shape. In the stationary phase where they reach the maximum population size, now we start to see high plasticity and differences in plasticity among our evolutionary treatments, which are these colors here. See? So the unpredictable treatment is this one. It stays low, whereas the predictable treatments, they get higher here. So lots of people now are actually interested in this, in the plasticity kind of subfield of what people really are becoming interested in understanding evolution of these rates of plasticity. How fast things change, how is this evolvable? Can you measure genetic variance in that? It's another piece of work. It's kind of complicated. Let's keep this.
Do you think this applies only to microorganisms?
No. I don't think it applies. What I think applies to microorganisms is that these are like one generation per day. So these trajectories across generations, I think it's specific to microorganisms because it's just mitosis. But the fact that there's this dynamic change, these lagging responses and things, I think it's quite common. And then across, in like larger organisms in plants, I mean, they do see these transgenerational effects when you study this transgenerational priming. You expose a plant to a stress or to a pathogen, and then you see responses in offspring in several generations. So unfortunately, I think it's quite common. I mean, because it doesn't make life easier.
I was just thinking about the later phase. This is, so if I would bring this to a sexual organism or whatever, yeah, this would maybe respond to what your exponential growth phase would be the young age, and then when you are aging, would you find then differences in plasticity? This,
well, age-specific effect probably. I mean, people. I mean, actually, we have studied that with my colleagues Anne Charmentier and Céline Teplitsky and a student. It was more, the idea was like senescence in plasticity, where it's kind of related. Is like, do older birds have difficulty in adjusting their breeding date? So for this type of traits, this is studied. People are interested in that. It's not exactly the same idea as this time to mount the plastic response. All these dynamics, I think they occur, and the problem is like when they start occurring on the time scale where it looks like evolution, it's cumulative, it's, you know, it's becoming a real difficulty, because you can say, okay, I'll just sequence everything and see if it coincides with frequency change, to say, oh no, it's not plasticity evolution. When it's both, it's kind of.
So the ultimate plasticity is behavior. And you might imagine in a natural population, you actually have a natural salt gradient in that water. May not be a lot, but, and then the algae could basically decide the trade-off between salinity and photosynthetic efficiency. So you probably won't get that in the lab because you probably don't have a deep enough water column to get that. But in nature, that might be one way they're handling that.
Exactly. I mean, there's a group in Moulis, in the Pyrenees, perhaps not far where Nick is sampling his flowers. They work a lot on this dispersal plasticity, which is kind of the same as habitat choice and all these things, but they have experiments where they're trying to understand how this works, and they found this in microbes, in Tetrahymena, so it's their whole object of study. Okay, so we've also studied plasticity of gene expression in this species. So we started by comparing our two preferred strains, let's say the red and the black. And so what we did is just place them in two salinities and compare their whole methylome, their whole genome bisulfite sequencing, and then their whole transcriptome. So, and for methylation, the response was a bit disappointing. There wasn't very drastic, there weren't drastic changes in methylation level across salinity. But it's a candidate that lots of people talk about, that this could mediate plasticity. There were still some differences among environments, but they were less marked than the difference among strains. For gene expression, there were clear-cut differences among salinities, and also genotype by interaction, genotype by environmental interaction of gene expression. So there's potential for genetic variation in gene expression plasticity. So the next thing we did was then to apply this to our evolved populations. And so we basically did the same, but we just picked, which Christian criticized, I remember last time, we just picked a few subsets of our lines because we couldn't sequence everything. So we picked a few of our lines and we did the same. We got their transcriptomes and methylomes. And so, I mean, I'm not very good at describing these types of graphs, but when you do the whole methylome and you do it's an RDA, redundancy analysis, like Christelle, I didn't show her, but she's a big fan of this kind of vegan package for multivariate analysis. So I wouldn't be able to explain exactly what redundancy analysis is. It's kind of multivariate regression with multiple responses and multiple explanatory variables, I think. So when you do that, you found that the methylomes are different across, there's different lines from these different predictability treatments in colors. It also differs among salinity. And there's an interaction between them. So, but if you look at differentially methylated regions, there weren't that many. And so we found a few that respond to salinity at low predictability, and then not the same. We didn't find the same in different treatments. For transcriptome, there was much stronger response, as I showed before. So there's a clear difference between these evolutionary treatments and their transcriptomes, and also they respond differently to salinity. I mean, they're not super exactly parallel. So it's not a very strong response. But when you compute this Euclidean distance between the whole methylome, basically, you see the change between high and low salinity at the methylome level, or the transcriptome level. And we did this against environmental predictability. We find same direction of the response to predictability as we found for cell morphology that I showed you before. So we remeasured this experiment. We found the same relationship, but when you look at transcriptome differences between salinity, they were also bigger or lower in this population that evolved in the low predictability. And same here for this DNA methylation, even though this is not entirely significant. So perhaps I should have used the combined p-values if I had known that the minus log of uniform is chi-square, but I didn't. So but now you do. Now I do. So now I need to retract this and do it again and show that there's a consistent pattern. If I do a test with all this together, this is what it means somehow. Is there do we find the same pattern across all these things? You would use these combined p-values, I guess? I don't know. Okay, I still have many slides, but I will end up with this. So we also looked at these tolerance curves that I showed you before, that where growth rate depends on previous and current salinity. And we looked at whether this evolves. Because you would expect, there's all this literature about environmental fluctuation should broaden the tolerance curve. You become less of a specialist, basically. We saw an effect. We also had constant treatments. We saw an effect of the constant treatment on the optimum salinity, but it wasn't very steep. But the ones that evolved at low constant salinity had a lower optimum than the ones that evolved at high salinity. What I found interesting is that it evolved in both directions, not just in terms of how growth rate depends on where you are, but also in terms of how growth rate depends on where you just have been. So for this, we did this whole array I'm not showing, where we have 9 salinities times 9 salinities, and we did all this growth. And when we compared constant to fluctuating treatments, so the constant is here in the background, the constant that corresponds to this, that has the same mean as the fluctuating. And so the fluctuating treatments, they had broader tolerance curves than the constant. Again, broader with respect to how the growth rate depends on current salinity, but also in terms of the effect of the previous salinity. So, so that's it. We were basically trying to bridge all these results about plasticity at different levels, at the genotype, at the phenotype, at the level of fitness, basically. So, but in the end, the tolerance curve, what we were expecting would be that fitness, if you were in environments that tend to covary in this way, then the whole thing would rotate, and how fitness depends on current and past environment would have rotated to match the level of the pattern of covariation in the environment. It's much too constrained to do that, so the shape didn't change much in response to environmental predictability. So I think I'm going, I don't know, I had a whole new section, but I think I'm going to skip it. I don't mind skipping it. I think, and I don't have a conclusion, so I prefer leaving it there and responding to questions if you have. We can also all go for lunch if you want. Or I can move to the third part. No. Yeah.
Can you please explain again why the plasticity is lowest during the exponential growth phase?
Sorry?
Why the plasticity is lowest during the exponential growth phase?
Why?
I didn't understand.
I don't have an explanation. It's just that we saw that you had these populations that, when populations are growing, that you have this exponential phase, and then they saturate, and then I'm just saying that I think you're referring to the fact that I'm saying people are measuring things here. People are doing, genotyping, RNA-seq, phenotyping. If you want, I have the impression, from what I've seen, that they usually work in this phase because if you have let's say you have different populations that are like this, in different environments, you want to homogenize them. If you take them at the same day, well, they will be in different, it's really different physiological stages. You know, in this high density, their whole metabolism is different. So usually one way to standardize it is to just take them in the exponential phase, because it's an easy standard, basically. You see? Just so that they're in the same physiological state, basically. But it just happens that for us, they weren't responding much to the environment, which makes sense because they're just investing everything into dividing a lot. That's my interpretation. Whereas in the stationary phase is where different things happen, they compete, there are limited resources, stress, perhaps they're more stressed. For instance, when they're at high density, this is where they produce the carotene, this is where lots of their responses that are known show up. So, but I think it's been seen in other microbial studies that have less diversity in the phases of rapid growth, basically. There's a stereotypical metabolism that corresponds to that and traits. Something else? Oh.